Dr. Sayani Bandyopadhyay. Today we will discuss about the principle of genetics. Genetics is a major discipline in biological science. So study of genetics is very important for all of us to know about our genetic disorders and the interactions of genes with the environments. The knowledge of genetics is very important. Introduction of genetics. It is a study of heredity and variation in organisms because we organisms have different genetic variations, and these variations is occurred due to the changes of DNA, RNA and protein. So the genes are the DNA sequences coding for inherited characteristics located in our chromosomes. We know that we have 23 pairs of chromosomes, where 22 pairs are autosomes and 2 are sex chromosomes. And the genome is a complete set of the genetic material. So Mendel, who is Mendel? We all know the name of the Mendel. So Gregor Johann Mendel. He is called as the father of genetics. He discovered some laws of genetics he established, and also he was working with the pea plants. Why with pea plants? Because pea plants have several advantages. They have total 7 different characters. You can see that the seed shape, round and wrinkled, seed color, yellow and green, pod shape, inflated and constricted, pod color, green and yellow, flower color, purple and white, flower location, axial and terminal, plant size means plant height, tall and short. So why did Mendel choose pea plants? Because the flowers are mainly bisexual. They have the male parts as well as the female parts. They follow the self-pollination process. The cross-pollination also sometimes occurs, so can be effortlessly conducted in some plants. And these plants have briefer than lifespan. So their lifespan is smaller. So lifespan smaller means the study time will take less time. The physical components of the plants are extremely simple and very easy to handle in the laboratory as well as in the field experiment. These are the terminologies of genetics. First, gene. It is a genetic factor that helps to determine the characteristics. And then allele, which is called as the alternative forms of genes. Then the locus, where it is present in a chromosome, where occupied by an allele. The genotype. Genotype means a set of alleles that individual possesses. And also phenotype means the physical appearance, what we can see. Heterozygous, an individual possessing 2 different alleles. We already heard about the capital T, small t in class 9 or 10. So the heterozygous, if these 2 alleles are different, means capital T, small t, capital or smaller, like that. And homozygous, if these 2 alleles are same, small t or capital T. So gene, we already heard that the gene is a segment of DNA that contains the instruction of making a specific protein. Allele, which is the alternative forms of genes. It can be dominant alleles. Dominant alleles suppresses the expression of another allele, which is known as recessive allele. So this is a monohybrid cross. Involves the inheritance of a single trait. You can say their trait is plant height. It is tall or dwarf. So if the trait number is single, then it is called monohybrid cross. If the trait numbers are more than 1, 2, then it is called dihybrid cross. You can see in this crossing, this is the round and yellow. So one is the shape, another one is the color, and another one is a wrinkle and green. So more than 2 different traits. So Mendel's law. At first there was a concept of dominance. Then the first law of segregation, and next the law of independent assortment. First, the concept of dominance. The concept of dominance states that the 2 different alleles are present in a genotype. So only the trait of a dominant allele is observed in the phenotype. Means here it is capital T, small t, and here it is capital T, small t, capital T, small t, capital T, small t, capital T, small t. So small t is for dwarf height and capital T is for is for long height. So tall. So capital T, small t. If these two alleles is present, then the phenotype will be tall. So the phenotype of that small t is dominated by the capital T. So only tall, not dwarf phenotype will be expressed. Then the law of segregation, which states that each individual diploid organism possesses two alleles for any particular characteristic. So it is based on the monohybrid cross. These two alleles segregate when the gametes are formed, and one allele goes into each gamete. So these two alleles segregate. That is why it is called as the law of segregation. So here it is a parental generation. Capital T, small t. And here it is a F1 generation, which is capital T, small t. Then it goes in F2, where you can see that 1 capital T, 2 capital T, small t, and T 1 small t. So 25% chance of having the progeny phenotype tall. 25 capital T means true tall. 50% chance will be heterozygous tall, and 25% chance will be dwarf. So the phenotypic ratio is 3:1. Tall is to dwarf. 3 tall, 1 dwarf. And the genotypic ratio 1 homozygous tall or true tall, 1:2:1, 2 heterozygous tall, and 1 homozygous dwarf. So 1:2:1. The phenotypic ratio is 3:1, what we can see, and genotypic ratio is 1:2:1. Then the law of independent assortment. It is based on the dihybrid cross. So Mendel actually discovered that this law, the law of independent assortment, which states that a pair of traits segregate independently, they are not dependent with each other, of another pair during gamete formation. As the individual heredity factors assort independently, different traits get equal opportunity to occur together. This is the cross. You can see this is the round and yellow. The shape, round shape, so it is round and color is yellow. Round shape is dominated over dintel shape, and yellow color is dominated over green color. So this is the dominant alleles and these are the recessive alleles. So you can see this based on the crossing, total 9 capital R, capital Y, capital 9 round and yellow was observed, and 3 round and wrinkle was observed, 3 yellow and wrinkle was observed, but only 1 yellow and green was observed. So the phenotypic ratio is 9:3:3:1. So genes on chromosomes. Genes are located on the chromosomes, and homologous chromosomes, the pairs carrying the same genes but different alleles. Genotype versus phenotype we already talked about. Genotype what is present in the gene, and phenotype what we can observe. So this is the chromosomes. There is a phenomenon called linkage. Linkage is the tendency that genes are closely located with each other. So gene is the tendency of genes located close together on the same chromosomes. So if these two genes are located in the same chromosome, so this can be called as linkage. And crossing over is the exchange of genetic material between two homologous chromosomes. This is the one chromosome, this is another. So this is the exchange of genetic material. So in this chromosome, some parts of this chromosome, second chromosome, and in the second chromosome, some parts of the first chromosome. So this is the recombination or crossing over. So genetic disorders. There are different types of genetic disorders. Either it can be autosomal disorder, or sex-linked disorder, or dominant disorder which passes from one generation to another, or recessive disorder. It skips the generation. We will talk about the 3 major genetic disorders and some chromosomal disorders. Thalassemia, which is an autosomal disorder, it produces lower content of hemoglobin. So it changes between in the genes. In hemoglobin, there are 2 types of hemoglobin. One is alpha globin and another one is beta globin. If any of the genes which code for the alpha globins mutated or deleted, or any of the genes which codes for beta globin is mutated or deleted, then abnormal hemoglobin content will generate, and this disease is called as thalassemia. Another one is the color blindness, which is the X-linked disorder. Means in female there is 2X and in male there is 1X. If in male 1X is affected and in female if 2X is affected, then this disease color blindness cause, as well as hemophilia, which is a blood disorder, and blood lacks a certain clotting factors, is also an X-linked disorder. These are the chromosomal disorders. If the numbers of chromosomes are changed, first one is Down syndrome. So it will cause an extra copy of the 21 chromosomes. Another one is the Edward syndrome, which causes the extra copy of 18 chromosomes. And another one is causing leukemia, which is the Philadelphia chromosomes, where no copy or no change, just chromosome 22 will be shorter. So in Down syndrome, there are separate facial appearance, distinct facial abnormals, developmental stages, different delays will occur, and in Edward syndrome, low birth weight, small abnormality in shape can be observed. And other sex chromosomal disorders are Klinefelter syndromes and Turner syndromes. In Klinefelter syndromes, the chromosome number will be 47. So it has one extra copy of X, XXY. For that, male born with Klinefelter syndrome may have low testosterone and reduce the muscular mass, facial hair and body hair. And in Turner syndrome, there is one copy of X chromosome is deleted. So the chromosome number is total 45 and one X chromosome. So symptoms: short, delayed puberty, short and infertility also occurred, and also some heart defect occurs due to the Turner syndromes. So for characterizing these types of disease, we have to identify which chromosomal disorder takes place. Thank you so much.